Hello, this is Dr. Jenkins. Here is our lab introduction video for the digestion lab. Folks, we are coming down to the end of the road. We're rounding third base. We have only 2 labs left worth of material. So that is good news. Keep working hard. We're not done yet, but we're almost there. All right, so here we go. We're going to continue with our discussion of body systems. We did the cardiovascular, we did the respiratory, and now we're switching gears to digestion. Make sure that you review all of this introductory material, but as I like to do, let's try and simplify it. Make sure that you can define digestion. Digestion means to break down. And that's what's the first step that we need to do when we eat something. We put something in our mouth, we begin to chew it, and that begins the process of digestion, which simply means to break something down. It makes sense, folks. We can't absorb into our bloodstream a big old honking piece of bagel, so we've got to break down that yummy bagel into its smallest pieces. And only then will we be able to absorb it. Now, there are 2 types of digestion. We have mechanical digestion and we have chemical digestion. So this really isn't that tough. We can break down foods using mechanical means. An example of that would be like chewing, or when the stomach churns. These are examples of how our body breaks down something using mechanical or physical means. And then chemical breakdown is when we break down foods using chemicals, more specifically using enzymes. And we must do both. So after we bring in food into our mouth, we will mechanically digest it. Then we will chemically digest it. And then third, we will absorb it. And that's what's listed right here. By the way, all this information is in this introduction. I'm just trying to simplify it. So the last thing we do is after something has been mechanically and chemically broken down into its smallest pieces, then we absorb that food, the smallest pieces of food, through the small intestine wall, because absorption only happens in the small intestine for most foods. And the key here is that we absorb the food through the small intestine wall into the bloodstream. Because we already know how are we going to be able to transport all these foods throughout the body. And the answer is we're able to transport them through the bloodstream. Lovely. All right, cool. There's quite a few videos to watch here, but they're not too long. So you're going to watch this video that talks about the digestive organs and their functions. It's actually a really cool 3-dimensional view, and it follows a piece of food through the digestive tract. So it's actually not so bad. And then based on that video, you are going to answer the following questions. So these come right from the video. Now, I want to say something. These questions, let me just say these. Number 1, you do have to know for the quiz. But numbers 2 through 4, you don't have to know for the quiz. I'm trying to give you some points here for watching the video, because this is going to be worth points towards your lab assignment grade. But I don't want to confuse you. These are things that you don't need to know for the quiz. But number 1, you do need to know for the quiz. All right, good stuff. Then I'm going to ask you to do some labeling. And you can see this right here. So just label the major organs. Then I'm going to ask you to fill in some of the functions. It looks like this kind of came up funny when I brought it over to this program. Here's the drawing, here's the table I was talking about. So you can see this table gives you a basic idea of the functions. It also labels it for you, so there's no reason why you should miss points on the labeling. But what I'm asking you to do, first you're going to just label, that's the anatomy. And then I'm asking you, and this is going to show up better on your own form there, I'm asking you to fill in the function. So the table looks like this. And you're going to fill in the function of the mouth, esophagus, stomach, small intestine. And for the small intestine, I want 2 functions. And then you're going to fill in the functions for the rectum, anus, liver, gallbladder. And we are missing the pancreas. There's the pancreas. We got 2 more. You're going to fill in the function for the large intestine and the pancreas. Use this table here to help you, this diagram, I should say. But I only want you to fill in the functions using these terms. I'm trying to simplify it. So when you fill out this table of the function, I want you to pick from this list: produces bile, chemical digestion, excretes poop, stores poop. And again, it's going to look better in the proper format. You may use some of these functions more than once. Not all of them, but you may use one more than once. Alrighty. Part 2 of this lab talks about enzymes. Now, at the very beginning, we reviewed some basic terms. Digestion is to break down, and we can digest something mechanically through chewing or the stomach turning, or we can break down something chemically. And when we break down something chemically, we are using some very important little proteins called enzymes. We wouldn't be able to digest things fast enough without them. We would die. The function of an enzyme is to catalyze or speed up chemical digestion. So it doesn't necessarily do the chemical digestion, but it speeds it up. Make sure you review some of the important characteristics of the enzymes. They are specific, and that means that each enzyme can only work on a specific nutrient or substrate. So for example, amylase can only work on starch. Amylase is the enzyme that can only help to break down or speed up the chemical digestion of starch. That's it. Amylase cannot work with fat or protein. Lipase is another enzyme. Lipase can only work on fats. So they work specifically. Each enzyme can only work on a specific nutrient. We can also see that after an enzyme does its thing to help speed up a chemical reaction, it leaves unchanged. And that's important because it means that the enzymes can be reused. That's a very beneficial thing. We like the fact that these enzymes can speed up chemical reaction, be reused, and so forth. Another thing, something that's a dead giveaway for knowing that something is an enzyme, is that it ends in the suffix ase. I've already given you some examples: amylase, protease, lipase. It continues on. Pretty much all of our enzymes end in ase. Acetylcholinesterase, creatine kinase means ACE, enzymes. Enzymes like to work at warm temperatures, so they get a little bit more active when it's warm. And this is one of the main reasons why our core temperature is warm, because it's the perfect environment for these little enzymes. However, if it gets too high of temperature, or if the blood becomes acidic, the enzymes will unravel. The fancy word for that is they denature. I like the word unravel because when you look on the video here for this one, which by the way is right here, when you look on the video, it's going to show you what an enzyme looks like. And it looks like all these squiggly lines. Well, when an enzyme denatures, all those squiggly lines are no longer clumped together. They literally unravel or they denature. And that can happen in really high temperature environments or acidic environments. And that's bad because when the enzymes denature, they can no longer do their job. All right, cool. You're going to watch the video and then you're going to answer these questions from the video. This one you will not need to know for the quiz. This one you will not need to know for the quiz. But these two you should know for the quiz. So you're going to fill them all out to get points on your assignment. But then as you're studying for the quiz, you only need to study what I'm pointing out. Alrighty, my friends, the last part of this lab is an experiment using saliva. Because guess what? Saliva contains an enzyme. Saliva contains salivary amylase, which, as we've already established, amylase is an enzyme that works on starch. So you're going to watch the video. And again, I'm sorry for the terrible music. If I made the video, it would have much cooler music. And then I've also written down step by step what they're doing in the experiment. They take starch, they're going to add it to water, then they're going to add that starch water solution to 2 different test tubes. Then to only test tube B, they're going to add saliva. You're going to be looking for a color change. At the very beginning, you can see that we're going to be adding iodine. Iodine is an indicator solution. When iodine is added, if that solution turns dark purple, it is positive for starch. All right, so your color key here is, if that test tube is dark purple, I think they say in the video violet. If it's dark purple in color, it's positive for starch. Any other color, it could be a lighter purple, it could be rainbow, it could be white, black, blue, red, yellow, anything. Any other color aside from dark purple is negative for starch. So when you watch the video, I'm going to be asking you to fill out this chart. So it's going to ask you the initial color, and you'll write down the initial color of each test tube, and then they're going to wait a little bit, and then you're going to write down the color again. And lastly, you're going to answer the question. All right, folks, keep up the hard work. If you have any questions, let me know. And as always, good luck.